Okay, good morning.
Good morning.
Not freezing cold in here. It's like everybody's first comment. Wow. Any questions before we get going? This is the stuff you'll need for today. You don't necessarily have to download the optional reading right now. I'll highlight that in the lecture notes later. And I'm hoping we get to the tutorial. I realized sort of after the fact this morning looking back through how many slides there are in this deck, but a lot of them are just transitions, so there's a lot of slides that build on each other. No questions about past stuff? So this week we are gonna build on what we've been doing, but move into aligning whole sequences. So for pairwise alignment, we'll look at some of the algorithms that have been developed. Ultimately, the one that is used by all software now, which is dynamic programming. Then we'll look at how you do multiple sequence alignment, and that's building on, or on Thursday we'll get to BLAST, which is a whole nother animal in terms of algorithms and heuristic searching. So why is sequence alignment so important? Fundamentally, the question is, what is the optimal alignment for those two sequences? Or any two sequences, and we're talking about pairwise. Anybody know why does alignment matter a lot?
Whenever you're comparing conservative parts of the genome to each other, or like you're just studying certain genes, you want to, usually in biological contexts, you kind of want things to be very similar whenever you're like doing tests and stuff like that, in like model organisms.
It's definitely helpful if they're similar. It makes the alignment easier. So you're definitely on the right track there. Yeah.
So if any like substitutions might be occurring, or there's one
Yeah, so I think you're sort of both on the same track there. Is it, how do you know if it's a substitution or a nucleotide change? Well, being able to interpret that depends on them being similar enough that you can confidently say this is the same position, right? So almost all downstream analyses, in if we're talking about sequence analysis, assume that when you show me the alignment or you analyze a certain position, that those are the same because of homology or common ancestry. When you get a set of sequences, that is an often implicit assumption of an alignment. So the examples here, variance between individuals, like if we're talking about human population sequencing, those differences at that position are polymorphisms in the genome. They're not alignment errors, okay? And that's relevant, right? Because you want to say that difference arose by mutation, not by an artifact of the way I aligned the sequences. If you're looking at fixed differences between species, we'll call those substitutions. In order for that to be interpretable in terms of what's the evolutionary cause of that difference between species, you're assuming that you've aligned it correctly, right? That it is actually due to selection or mutation and then selection or drift that has made that site different between species. It's not an artifact of the alignment. Okay. So that is all to say this is a very important biological question, and it is a very big problem computationally. Okay. So how does this differ from what we've been doing so far with sort of searching for patterns? Just intuitively, not asking for algorithm details. Somebody take a stab.
Are we taking like maybe larger sections and just kind of like trying to align them instead of just taking sections and kind of find repeating patterns within the sections?
Yeah, exactly. Right? Instead of looking at us trying to find out where this little piece belongs in this bigger sequence, we're going to take two whole big sequences and try to figure out what's the best way to align the whole thing of those two big things, right? So that's a different computational problem than pattern matching, okay? So the alignment game, this is, I took that title straight out of the textbook, that Intro to Algorithms textbook. The game is to maximize the number of points, and you get a point if you match at the position, right? So if you think about how we align these two sequences, you get a point because the first position is a match. You get another point because that one is a match. Then we get to this situation where there are three possibilities. The bottom one there is, right, the original, where that's a GC and we're going to give it a 0. Two other possibilities is we could insert a little gap there. So the dashes are gaps in the sequence. And it's basically just shifting it to see if we can get a better alignment if we introduce that gap. So in this case, you could introduce a gap in the top sequence or the bottom sequence. In either case, it's going to score a 0 at that position. So for the third position, all three of these options are equally likely. If we move to the fourth position then, so I've drawn this out so that we don't make any assumption about what was correct in the first one. Now you can see, okay, if we put this gap in the second sequence right here, there are two possibilities for position 4. Well, really 3. You can have the TC, which is still a mismatch, so that's a 0. You can insert a gap on the bottom, insert a gap on the top. All three of those are still going to give you 0 for a score. For the one on the bottom, right, now at position 4 we have TG, which is still a mismatch. You could introduce a new gap. Again, that's going to score 0. So here, 3 and 4, both positions are still scoring 0. On the top one, when we inserted a gap here, that gave us a match at position 4. So you might say, okay, if this sort of simple algorithm is working where we just move step by step, you've got a good candidate for position 3 having a gap in that top sequence. And then you just keep sliding along. Okay, so I've gotten rid of the two that had a lower score after 4 positions. At the 5th position, we get another match. You start to really, you can hopefully intuitively see even here when you start to try to explore all possible scores, like I at every position I could insert a gap in the top or the bottom sequence or leave it alone. When we start getting out to position 6, 7, 8, all these scores are 0, and if you want to draw it out just while you're taking your own notes to think about how this works, it doesn't really matter what you do in those last 4 positions. There are a lot of possibilities that give you the same score, which means there's no real way to know what's the correct one. So this is why when I said this is an important biological question, but a hard computational problem. There is no computational solution to this. All 4 of those sites, if this is all the information you have, there are multiple ways that you can align those last 4 sites and they all give you the same 0 score. Okay? Yeah.
In the lab, how would you apply this to, in the lab? Like, would that be damaged or missing sections, the spaces, the lines?
Very good question. How do you interpret those? What those mean, so how do you read this sequence alignment? Well, matches, that's easy, and I've sort of drawn it out in the way that you often see it, where these little pipe characters are connecting the matches. In this simple 2-sequence example, I'm naming insertions and deletions relative to sequence 2. So this gap in the top sequence is an insertion in sequence 1. Okay? Now, what's the difference between an insertion and a deletion? Well, it's which one did it happen to. When you only have 2 sequences, you don't know, right? So what I'm saying is, at that position, if all I have are these 2 sequences, I could call this an insertion in sequence 2 or a deletion in sequence 1, right? And same thing when I slide out here, I've called these deletions in sequence 2, but you could call them insertions in sequence 1. You don't know the polarity of insertion deletion when you only have 2 to compare to. Okay. Does that answer your question?
It does.
And in case I forget to say it explicitly later on, because all 4 of these sites kind of have equal probability, this is a section that doesn't align well, and if you're doing some downstream analysis, you wouldn't want to put a lot of weight in your confidence of homology for those sites, right? Okay. So, in this little cartoon example, how you find the optimal sequence alignment, it seemed simple, but it's partly because I didn't draw out all the possible alternatives for those last 4 sites, right? So if we really wanted to explore the total number of possible alignments for these 2 sequences, this is how it scales. And that, if you haven't had a probability class, that's okay. This says 2n choose n. And so that's 2n factorial divided by n factorial. That is a really big number. Okay. So in a nutshell, the computational cost of exploring all possible alignments scales exponentially. So when we talk about like brute force alignment, if you get much past 10, 20 sequences, this is a huge number. You really can't explore all possible combinations for an alignment. Especially, we'll talk about this, only gets worse when you talk about multiple sequence alignment. So, computer scientists to the rescue. They know this problem called the Manhattan tourist problem. It's called the Manhattan tourist problem because this is like a cartoon map of Manhattan, And the problem is find the optimal path, where the optimal path is a walk through Manhattan where you only go south and east, but you visit the most attractions possible. So how can you get through Manhattan and see the most interesting stuff? Okay. So we're walking from the origin, which is the blue circle, to the destination. Each one of these little black squares is an attraction. And so how do you get from one side to the other? Well, you can simplify that into a graph. And so here, all of the edges that have an attraction have been highlighted. So all of these dark arrows that have a 1 mean if you go across, like if you take that street across that block, you're going to get to see one thing. Okay. You can see there's only really one block down here where you get to see 2 attractions on in one block. And so now, how do you find what we call the longest path? And we call it the longest because it has the highest score. In this case, the most attractions. In case the terminology is not clear, nodes are the dots, edges are the lines in between them. Okay? And so the edges are weighted by the number of attractions in this Manhattan question. And if you stare at it for a while, draw the arrows, try to find the best route, okay, you will find that the longest path is if you can only go south and east, right? You can only take 2 kinds of turns, okay? This is the optimal path. So at most, if you can only go south and east, you can visit 5 attractions. Well, it turns out that you can formulate the Manhattan tourist problem as the optimal sequence alignment problem. So you can think about having the sequences in an array, like a 2D array, and we're going to score the attractions, in this sense, as matches, right? If you match going across that position, then you get a 1, otherwise you get a 0, okay? And so if you now think that is like the Manhattan problem, how do you design an algorithm to walk the optimal path, right? Because a second ago with my yellow lines, I was just eyeballing it, right? Like mentally looking at the map and seeing what's my optimal path. But I have to be able to tell a computer to do that somehow, okay? So now I've redefined the edges in this map, so this isn't the Manhattan attractions, but all of those, so the grid numbers are across the top, down the side, and then the edges have all the weights, okay? There are lots of possible paths. So this is just like I was saying with our cartoon DNA alignment. If we had to enumerate every possible alignment, that number gets really big really fast, okay? Same thing with traversing this little cartoon Manhattan. If you have to trace all possible paths, that's a lot of paths to trace, and we'll see when we get to the recursive algorithms. That's the only way to find the true optimum. But are there simpler, faster ways to do it? That's what we're looking for. Option 1, called greedy algorithms. Okay. And so what that means is from every node, you're going to take the edge with the maximum value. So, right, in that case, so we've got 2 options. From the origin node, we can take an edge that is a score of 3 or a score of 1. Okay, we take the one that says 3. From there, we take a 0 or a 2, take the 5. You get the idea, right? at every node, we just take the edge that gives us the better score. Okay, and if you keep doing that, you end up at the origin, and what we would call the longest path has a score of 23. So if we're using the greedy algorithm as our faster solution to solving the tourist problem or finding the optimal alignment, the question is then, does it work? Is 23 the longest path you could visit through this version of Manhattan? Okay. Well, back up a step to the computational question again. Do greedy algorithms work? Why is this a thing? Well, they do work, but the situations where you can be sure that they are the optimal solution are pretty limited. The classic example is, how do you make change? And like, anybody ever think why we have the denominations that we do? Well, economists aren't dumb. They know these things too. And so the US coin system, it's not the same as the European system, but they will both, in this case, I'm just giving an example of 40 cents, but I think this works for all possible denominations, where the coin, the optimal number, works using the greedy algorithm. So think, how would you make change for 40 cents? You would give them a quarter, a dime, and a nickel, right? That is the fewest coins you can give a person and make 40 cents. It works with European coins as well. They get it more efficiently because they've got a 20-cent piece. So they can give you 2 coins. Where it doesn't work is there's a, like Tanzania has a 25 and a 20-cent coin. I'm not sure they were all minted in the same year. Like, I think they might just be all in circulation and they changed at some point. But that's neither here nor there. The problem is the greedy algorithm will give you the same solution as the US coin system, but that is not optimal for the currency in circulation in Tanzania. The optimal solution would be give 2 20s. Okay? So why is it greedy? Again, you take the largest coin that doesn't go over the value, and that's a 25-cent one. Then once you've given the 25 cent, you're stuck giving the 10 and the 5. Otherwise, it will go over. So that's a sort of fun example for why greedy doesn't always work. And so applying a greedy algorithm to our tourist problem or our alignment problem is not guaranteed to give you the optimal solution. And so here I've drawn another solution that gives you a score of 34 working through that. So this is not a good option in terms of algorithms because it's not guaranteed to give us the longest path. Okay, so if not greedy, we can toss that one out. It's fast. Greedy will give you a almost instant solution, but it's not going to be the optimal one every time. So second one is the recursive algorithm. So these are super cool, and I remember the first time exposed to recursive algorithms, it kind of blew my mind. This is like, I don't know, the multiverse in my mind. So these are algorithms that break the problem up into a fundamental piece that allows you to just call the program itself. So in this case, our function is going to be called the minimum number of coins. And to solve the problem of how do you make change, you're going to have that function call itself. So we're going to pretend we've got denominations of 6, 5, and 1, and we need to make 9 cents change. I've made the number small to hopefully, I feel like the algorithm itself is mind-bending enough. So we got simple numbers. To make 9 cents change, intuitively you would say if I've got 6, 5, and 1, I'm going to give a 6, a 1, and a 1, okay? But how do you tell the computer that? Well, our function, minimum number of coins, and we'll say we need to get 9 cents, is get the minimum of calling this same function, which gets the minimum, on 9 minus 6, which would be if I gave it a 6-cent coin first, or 9 minus 5, or 9 minus 1. Okay? So to get the minimum of 9, I need to get the minimum of all three of these options. Right? And so what I have circled there is what that ends up feeding into is get the minimum coins for 3, 4, and 8. Okay, so then the question would be, okay, I gave it 6. Now I've got to solve min coins 3. So I feed that in. And you can limit the search a little bit to requiring positive numbers when you do that first little math to say what's next. So if I gave it 6 cents, now I've got to solve min coins 3. 3 minus 1 is 2. Any of the other ones I can throw out because they're going to be less than 1 or less than 0. So min coin 6, this round says 1 cent. And then if I feed that back in min coins 2, I'm not drawing out min coins 2, but what is min coins 2 going to do? Just intuitively, like if you were going to fill the slide in completely, what would that look like? This is audience participation time. So this line is saying to get the minimum for 3 cents, the only option here is 3 minus 1. So if I'm now saying what's the min for 2, right, the only option is going to be 2 minus 1, right, and which is going to then result in me needing to go find min coins 1, right? And so then it's finally only when we get to min coins 1, there's no other option. That just gets a value of 1. Okay? And now, and think about what's happening in the computer here. It hasn't been able to solve this first step because it had to call itself to go get what the minimum for the lower change values are. Then it had to go do that again, still no answer. Then it had to go do it again to get to 1. Finally, it has an answer, and it repopulates all the way back up to the count of basically how many iterations, how many recursions did I have to do. That's what the little +1 is doing every time. So it's adding 1 to this value that says how many coins have I given it. The reason I've highlighted min coins 3 here is just to draw your attention to the fact that we've had to recompute the same min coins function 3 times already in 2 levels. And so this algorithm is guaranteed to give you the optimal change. If I extended this to the Manhattan plot, the Manhattan function, where now I've got to do minimum steps to get through that grid, it's guaranteed to give you the correct grid, like the correct longest path. But it has to compute every single possible path. So this, for the recursive tree, is why we don't use this method. So here, I need to redo this just so that all the numbers are the same, but like here I've given you one for making 76 cents change, okay? The optimal coin combination for 69 cents already by this level, and I just got tired of drawing things in, is 5 times. If you scale this down, and this is 76 given our 651 denominations, by the time we get down to where we're at min coins for 30 cents, we've had to compute that min coin function 1 trillion times, okay? Like this, because this tree gets so crazy so fast. Like, think you've got to count down from 76 by ones. Like there's at least 76 potential layers where there are at least 3 values at every one. Okay, that gets really big really fast. So doing that on any meaningful length sequences to get the optimal alignment becomes not doable. Okay. So this would work, the recursive algorithm. It's guaranteed to give the optimal path, but it's too slow. And what this really great mathematician discovered, and he's on the next slide, is that with a small tweak, we could not have to do all those computations and still use the fundamentals of the recursive algorithm. So he said, instead of doing all those time-consuming calls where you're computing and recalling the same function, what if you just keep track of every cell value so that you only had to compute that, like the min coins 3, you only had to compute it once, and then you just look it up, okay? And that's fundamentally what dynamic programming does. So here, what this is doing is working backward from 0. If we've got a 1 cent coin, to get 1 cent, 2 cents, 3 cents, 4 cents, all of those just take the 1 cent coin. Once you get to 5, now the value of how many coins is 1 again, right? Because min coins for 5 is just the 5 cent coin. Same thing for the 6 cent coin, okay? And so now we are remembering or storing in memory the minimum number of coins it takes to get to, to whatever value we need to get to. And so this guy, Richard Bellman, is the one, that is the dynamic programming algorithm. We'll talk about a little bit more in detail on how it applies to our alignment problem. But there's just cool history here. The dynamic programming really has nothing to do with programming. This is sort of interesting. Just he was working on an Air Force project. I can't even remember off the top of my head what it was for. Something with logistics. But like his money came from Secretary of Defense, and he just wanted a name that was more likely to keep him funded, and so he called it dynamic programming because he thought that sounded sexy. Sexy enough to get funded. Yeah, so planning. So something with logistics. Planning's not a good word for a lot of reasons. So anyway, something not even a congressman could object to. Okay, so how does this apply to our longest path problem? We can only move south and east, right? So we gotta end up moving into this by either moving down or to the right. This is what the algorithm looks like, okay? So if we start out at n and m is 0, that's the node up in the top corner. So that's just gonna be a starting value of 0. That's all the first if does. Then, as we move n greater than 0, right, moving down or to the right, if we go n minus 1, then we're moving down, plus the weight of how valuable is that edge, right? Instead of just adding 1 like we were with the change problem in the terms of numbers of levels, we're going to score this by how valuable is that edge. Should I move down or should I move to the right? Okay. Y, we're just saying if you move to the right, what's the weight of that edge? And then you return the maximum of that value. This negative infinity is just a way to make sure you don't stand still. Okay. You're not going to have to code this, so that's why I went through the algorithm kind of fast. And we're not doing an example to try to code dynamic programming. So here, going to walk through this, right? In that case, this node would get a value of 1 because the edge weight, if you applied that algorithm that I just was showing, the value of that node is 1, okay? Moving down again, the value of that node is 5, okay? And hopefully there's a mental link here with what we were doing with the coin problem, right? Instead of just filling out that row in the minimum number of coins, now I'm filling out the nodes in this grid, but it's basically the same thing, okay? And so I just jumped ahead, filled in the node values. Now we can start to fill in the internal nodes. And so here, that's what that max function, like when you return the max, like how do I get here? I could arrive by the top one coming down or the left one coming right. And so I need to say, well, which one gives me the higher score to get to that node? Well, it turns out coming down and then going to the right gets me a 4 at that node. Okay, so I've just bolded that edge to say that's how I got to that cell. And then you end up doing the same thing. Which direction gives me the highest score going into every node? And so here, filling out the rest of this and keeping track at every stage, which is the turn I made to get to that score. Okay, and there the whole table is filled out. And you can see, if you compare it to my earlier slide, 34 is the maximum, like the optimal path, the longest path through this city, okay? And I got that just applying that algorithm that was on the other page, right, a couple slides ago. So now the question is, how do I backtrack through this to figure out what's the alignment? So they're called backtrack pointers. So as I was populating this grid, I was keeping track of, to get to 32, I actually went east, okay? That's the interpretation anyway. Yep.
I have a question about how you got to 34 here. So a few slides ago, when we were talking about the coins, you said that the greedy path is not always the optimal path, but sometimes can it be both?
It can, but under very limited circumstances, so in this city, greedy won't get you to the maximum, right?
Okay.
Greedy got us, think about, I mean, you can go back in the notes and actually do the greedy path. Actually, I think I drew that. Right? If you took the greedy path at every edge, you would travel this way and only get to 23.
Oh, okay.
And if you did the recursion, you would have to do all possible values, like what are the possible values for that node right there? It could be a 4, could go 1 and then plus 3, or it could be a 3. Okay, and so think I've got to now my possible things that I need to consider. I have to hang on to the 3 and the 4 and do the whole tree out from 3 and 4. Okay, and so like once you get down here, you've got multiple potentials at every node, and that's what that tree, right? That's what's happening here. Okay, you've got multiple potentials at any given node, and the tree just expands every time you've got multiple potentials. Okay, so dynamic programming sort of gave us a way to only store the maximum at every node. It said, actually look and see, whoop, I went too far. Look and see, how did I get to to 4 here, and only store that, right? That's my backtrack pointers. Only store that I went down and then over. It's a good question. This is a lot, I know, when somebody's just talking at you. This will take going and looking back at it. Like, I get it. Those backtrack pointers, though, that we've stored give you this record of going through not the maximum edge, but the maximum node value at every step, okay? And so this gives us the optimal solution, just like recursion did, but now on a time scale that is computationally feasible, okay? This was solved by Needleman and Wunsch. This is like one of the most famous papers in bioinformatics, because this is what made sequence alignment possible. And this is the pairwise solution. So just looking at 2 sequences, how can we align 2 sequences? So what's the connection between Manhattan Tourist and the alignment game, right? I've already sort of given you a glimpse. I've showed you that it's the same problem, but here instead of giving +1 and 0, I've sort of given a indication for like what does that look like moving through the city. What we're going to introduce and what Needleman Wunsch introduced is this diagonal that moves you along both sequences. Right, so here's my Manhattan with the sequences on them, and then what I've done is shown all the possible matches where we're going to go on a diagonal. And so here, anytime you cross one of those red arrows, you get a +1. Anything else is a 0. And so we can apply the dynamic programming kind of algorithm to find the max at every node, whether we move down, right, or diagonal. And this is just so here, weight of the edge, I filled it in as 0s and 1s. That's what I was saying before. You move on a red arrow, you get a 1. If you move on a 0, on a right or a left, you get a 0. What are the right and left in terms of biology? I've color-coded them on purpose. Yeah, insertions and deletions. If you have to move right or left, that means there's not a match there, and so you're going to scoot. That's where you insert a dash when you're actually looking at the alignment, okay? And so that's why you're going to give them a 0. The red are the matches where you get to just move one down on both sequence axes. Okay, and man, the fonts really got messed up here. So if we applied our algorithm to this and kept track of the backtrack pointers, what is the optimal solution? Well, okay, so here I've filled it in. And when you have a scoring matrix that is 0 and 1, you have lots of possibilities to get to the same, like all of these black arrows that are backtrack pointers saying, I got to the same score. That's because they're all +0, right? It's not changing the score, and so those directions are all equally likely. But the optimal path, right, the one that gives you the best value down here, is what I've traced here, which is color-coded to the alignment you have over here. And something to notice, right? I said at the beginning that there's no way to know the right alignment for those last 4 positions. You can see down here when we get into those last 4 positions, all of these paths are equally likely, right? They all have backtrack pointers because they don't change the value of the alignment, okay? And so it's somewhat arbitrary that I've drawn those purple greens on those last 4, okay? That is one. Real code, when this really happens, it depends on the software you're using, but you can think of it as just random, okay? It's just picking one, okay? Right? So that's dynamic programming. And then the important thing to store, like for future reference, when we talk about BLAST next on Thursday, is the scoring matrix. Right. How might you get a better differentiation among the paths? Because there are so many equally likely paths there. Okay. Well, this is the scoring function we're using right now, where all the matches are a 1, and any other, like, so that's why the diagonal here is a 1. Those are the exact matches. The minus mu are if you have a nucleotide mismatch, then you've got these sigmas where that's a dash. So if you have to do an insertion in one or the other, in our example, we've given them all zeros. But you could do something like this, where a mismatch between nucleotides is less likely, you give it a more negative score than an insertion deletion, okay? So in this case, we're scoring mismatches between nucleotides slightly more strongly than we're penalizing the indels. But that's just one example, okay? And if you applied this scoring matrix to this sequence, you would get a score of -8, okay? Yeah.
And so, whenever we're using this, like, in bioinformatics and as a biologist, would the end result of, like, the scoring matrix be, like, a higher percentage of, like, matching? Or, like, so, like, say we're comparing 2 genes, and, like, this gene in monkeys and humans has a higher percentage of matching versus this gene in zebrafish. Is this where the scoring alignment comes from? Like, that's number?
That's the kind of question you should be asking. I'm not going to answer it right now because we're going to get there and it's a complicated answer. How to score based on how similar you expect them to be is a good question, especially when it comes to gaps. So, couple steps before we get to the answer to that. First, DNA. We know not all DNA mismatches occur at the same frequency, right? We talked about C to T transitions already, and like when you deaminate cytosine, turns into a thymine, okay? You see those way more frequently. So in your scoring matrix, you might want to say the C to T mismatch shouldn't be penalized that much because it might be a legit alignment, okay? We also know that, say, third positions in the codon, they code for the same amino acid. So we know they're more variable. They're more likely because the mutation in that third position doesn't affect the protein sequence. And so maybe third positions you want to weight differently, okay? You can take the genome frequency of a nucleotide into account, right? If you know you have a really GC-rich genome, you might want to make the weight of mismatches in GC lower, okay? the last thing I want to note, because all of my examples have been in DNA context or RNA, DNA and RNA have inherently less information than amino acid sequence. Why is that? This is not really a biology question as much as a analytics question. Why is there less information in DNA sequence than protein sequence?
Well, does it have to do with there being more like combinations and possibilities for an amino acid sequence?
Why are there more combinations?
Because there's like 16 amino acids and only 4 nucleotides.
That's exactly right, except there's more than 16. Aren't there 23? No, 20, 22? Okay. There's some modified ones too. So, in any case, the fundamental answer is correct. The alphabet size, right? DNA has a 4-letter alphabet, and protein has a 20-something letter alphabet. Okay. And so that is, from an information analysis perspective, way more information in a protein sequence than in a DNA sequence.
And then even though you can transfer DNA directly to the protein and get effectively the same thing?
Yeah, but yes. And so when I mean there's more information, so for alignment and homology inference, think about what are the chances of making a match in DNA sequence just by random chance. It's 1 in 4 versus 1 in 20 if you're talking about amino acids. So way less likely to have a random match. Okay, so going back to this, how should things be scored? One of the early efforts to say, how do we score mismatches in protein sequence was the PAM matrix. This did alignments of some well understood, this is back in the 70s, so there weren't that many, right? But these proteins had been sequenced. They aligned them and then said, okay, how often do we see the actual substitutions happen in these proteins? And so, right, think of it like this grid where these are all the amino acids down the left and across the bottom. And this is the PAM 250 matrix. So I'll get back to that in a second. But the weights here say how bad are we going to penalize 2 amino acids if we see them in the alignment. So here, right, tyrosine often mutates to, I don't know what F is, okay? So it actually gets a score of 7. So if you lined up a Y and an F, it would be +7 in your alignment algorithm. Very rarely do you see a Y and a P line up in that set of proteins that make up the PAM matrix. So you're going to penalize that one pretty heavily. So PAM1 is like the actual numbers of times, and then to get things, this is back to the question of human versus chimp, human versus zebrafish versus Arabidopsis, fly, whatever. The farther away you go, the higher the PAM matrix you might want to use. Because this is being raised to some exponent, which means it's essentially flattening the distribution of scores. And so the farther apart they are, the less you want to penalize or reward the mismatches, because over evolutionary time you expect them to be more divergent. And so I guess maybe it's easier to think of in things that are closely related. If they're closely related and there's a mismatch, you want to penalize that really hard, okay? And so you would use a low value PAM matrix. PAM 250 gets used the most because it's a happy intermediate if you don't want to make the assumption of how similar do I expect these sequences to be. Okay. The reason I say it's a good choice for evolutionarily similar sequences is that, right, this is based on literal alignment where you only know, and I think it was all mammals, okay? So it was trained on sequences that are pretty similar to each other. So it's really best for using as weights for things that are pretty similar to each other. As you raise it to an exponent and flatten out that curve, you're extrapolating based on no biology, okay? So yes, it will work for that conceptual model that I was saying. Farther apart, we want to put less weight on the differences, but we don't know if that's true or not, right? You may actually, even at long distances, never get this kind of mutation because it changes polarity or hydrophobicity or something like that, okay? The other scoring matrix that you see super commonly is called BLOSSOM. This was derived slightly differently. It's better for more distantly related sequences. I think we're running out of time. Conceptually, they were doing basically the same thing in that they took there were more sequences by the time this was derived, and it's just rather than extrapolating by raising the matrix to a power, they looked at how evolutionarily similar are the two sequences and recalculated the matrix based on those sequences. So, for example, Blossom 62 are only using the proteins that are 62% identical make the scoring matrix. Blossom 100 would be, okay, only the things that are 100% identical, now what's the scoring matrix, okay? That doesn't actually work because there's no changes. There are no mismatches in 100% identical. You get the idea. That was right. So a lower number in Blossom is better for more distantly related things. So the PAM numbers and the Blossom numbers go in opposite directions. That's the important thing to remember for possum. PAM and Blossom, that was the conflation of those two things. Okay, so higher numbers in Blossom, better for closely related. Higher numbers in PAM, better for distantly related. And so this was just showing how does that apply to our algorithm that I was showing. So we just have to add some additional weights in terms of how do you calculate the edges there. Haven't changed anything in principle, right? The matches still get a +1. You get to assign some different score for insertions and deletions, and then mismatches if you want to score those differently. Okay, getting toward the end of the conceptual stuff. Which of these two alignments is better? I'm not expecting you to do the dynamic programming in your head to give me a number, but which you think the first one? Why the first one? Has a higher score, yeah, by our match mismatch. Anybody want? Yeah, Manny.
More matches across large sections of the DNA as opposed to just like this one sequence and a bunch of scattered ones.
Yeah. These are very different looking alignments. And so the question is, in terms of the biology, which one is better in the sense of which one do I believe is showing me something about homology? The second one? Okay, this is yeah.
The bottom one has more of a continuous longer fragment that matches.
Yeah. It's the longest common subsequence in computational jargon. The real answer to this is you can't tell computationally. Okay, most biologists would probably say the alignment 2 it tells me something about biology. That is a conserved domain, right? Maybe it's a DNA binding domain, something like that. Okay, so this is the classic example: homeobox genes or Hox genes. They contain a DNA binding domain that is conserved across all animals, basically. But you can, like, if you try to align the genes, they have diverged a ton. Okay, you can only identify them by the DNA binding domain. Okay. So in many cases. Okay, so that local alignment tells you potentially something about function. Probably those sites are homologous, and right, it comes with lots of caveats about it would take actual experimental testing to know. So what does that look like in a big alignment? Where, again, now I've drawn the graph where I've got a sequence across the top and the left. The global alignment, so the one that gave the alignment 1 in the previous slide, is down there on the bottom. The second one is the top alignment. So this showing the best local alignment. And so how you get that is by weighting the insertions and deletions differently. That's one way to do it. Okay. So the local alignment versus the global alignment. This was solved by Smith and Waterman, who basically said if you drop below some threshold, you're just going to score it as 0 and start over. That's it in a nutshell. So it added this fourth option to the recursion rule that if you go negative in terms of your score, just stop trying to align and start over in a new spot. And what that ends up doing, this is my cartoon version, is it gets you little regions like that, but then it will go start over and start trying to align in other regions of those two sequences and see if it can get another good local alignment. Okay. And so that, like I said, comes down to how do you want to score the gaps? Up to now, we've just been saying any dash you have to put in there, you're going to score as a 0. But maybe 2 gaps, like inserting 2 separate insertions, is like that's less probable than having one insertion that just moved it 2 spots. Okay. And this is it's called affine gap penalty, where the gap opening and the gap extension have different weights. Okay. Almost always the gap opening gets penalized more than the extension. So it's more costly to do this, right? Opening 2 gaps is penalized more than opening 1 gap, but extending it. And this is mostly from biology intuition. Right? thinking about the number of mutations it takes to get to a certain sequence. It takes one mutation to get at least Occam's razor, the simplest explanation for that 2-gap insertion, one mutation of 2 nucleotides, or one deletion of 2 nucleotides, versus 2 separate deletions or 2 separate insertions. The one event is more likely. Okay. This has been a whole field unto itself of trying to optimize gap penalties. I'm not going to read this slide to you, but it's worth knowing, and almost all software has a way for you to tune these. And you tune them because, back to the question of the similarity of 2 sequences and how do you score, you might want to use different gap penalties for human versus chimp or human versus fly. Okay. And then the last thing on there is something to keep in mind. Getting the best alignment, sometimes a matter of trial and error. Okay, and you always end up with a hypothesis for the alignment. Okay, I'm not going to get into, so that's really the nitty-gritty of how does pairwise alignment work. Okay, for multiple sequence alignment, we're not going to get that far in the weeds because it's all based on that stuff. So it's just, how do you apply it? How do you apply the dynamic programming to 3 sequences, okay, or thousands of sequences? Becomes a hard problem. This is the generic workflow. We're going to go through the first 3, not today, but hopefully by the end of Thursday we'll get through these. So you'll get that data. We're going to work with that. Probably end up having to move it to Thursday. So you would get your sequences. In this case, I'm giving you the sequences. And then your alignment. These are the 3 main ones that get used: MAFT, muscle, and Clustall. There are lots of multiple sequence alignment programs. I'm giving you a link to the European Bioinformatics page where they sort of keep a catalog of the most commonly used ones. This is a current paper, so back in Canvas I had the optional reading. This is the most recent review for multiple sequence alignment. It's interesting. This quote that I pulled out: machine learning is valuable achievements in many fields of bioinformatics, but there is more work to be done across MSA. That's their politically correct way of saying machine learning hasn't really contributed much to our ability to align multiple sequences. So these approaches down at the bottom, really not better or faster than the classics, which are only 10 or 15 years old, but Clustall, MAFT, and muscle are still the ones you see in the literature all the time. But active area of research, because it's, again, super biologically important question to get the right alignment. And so there's a lot of people working on this and how to apply modern machine learning methods to the problem. In practice, which one do you use? The reason MAFT, Clustall, and muscle to some extent get used is because they're fast and they do good enough. Muscle, I would say, on smaller data sets still gets used pretty rapidly because it's quite accurate. If you have thousands of sequences, MAFT and Clustall Omega get used. I would say MAFT and Clustall Omega are the 2 most common that I see in papers that I'm reading. Okay. One of the cool things about MAFT is that it tries to figure out the optimal method for you. It first looks at your sequence. How much sequence did you give me? How long are they? and then I'm going to use the most accurate method that will complete in a reasonable time. Okay. So math is this whole family, but you can see it's like this is the phylogeny of how these methods are related. It's like a branch off of Clustal, but tea coffee and muscle are also very similar branches because they all are based on these two innovations. Clustal kind of paved the way for progressive alignment, which I'll show in a second what is progressive alignment. And then the second one that MAFT and muscle in particular iterated on or evolved is I said iterated on. It's the iterative and consistency methods. Okay, and again, I'll explain what those mean in a second. And then since then, like over the last decade or so, it's really just been computer science ways of getting these things to run faster. Okay, not just faster hardware, but MAFT, the two F's are for fast Fourier transformation. It's just a way to index the sequences to make it run faster on the computer. If you look at the scalability of these things, so we've got how many sequences versus how much time does it take to align them. What are the... Oh, it's the dots are the average sequence length. So big dots are longer sequences. Note these are both on log scale. So you can run muscle under two different modes, which is why the green is up at the top, meaning it takes a little bit longer to run muscle in its, like, give me the optimal sequence alignment. It goes slower. You can switch it into a faster heuristic mode, which will drop it down into the realm of the other ones. The reason MAFT and Clustal get used the most is you can see they're like down here in terms of how many sequences and how much time. They're the fastest. They give you the best answers. Just to put that in sort of tabular format, the compute time in seconds over on the right, and the score, higher is better. So they're not too different for most of the modes that you can run them in, but the time is really different. So progressive, the first major advance was like, and this is the first step in all of the modern sequence aligners, is align all five, do a fast, most of them use k-mers, like what's the nucleotide distribution basically? You know what k-mers are. It splits it into k-mers and counts how many of every kind of k-mer there are, and then makes a distance matrix. How many k-mers do they share for all pairwise comparisons across the sequences? Then you build like a phylogeny. They call it a guide tree based on how similar the sequences are or how many k-mers they share. Then you go back and start aligning. So if this is the guide tree, C and D are closely related and A and B are, I say closely related, they're similar sequences. So you align A and B, you align A and D, then you make a consensus for C and D. So now we've got our two alignments. We say, okay, what was the ancestor? What's the consensus of those two? Do the same thing for A and B. So now you've got these jagged lines are meant to represent the consensus sequence. Now you align those two consensus sequences. So now, if you think about what's happening on the tree, we've aligned these two and made a consensus. That's this branch right here. The C and D alignment consensus is here. Now we're going to align those two things. Whoops. To get this one, right? And so the last step is we've got our consensus of CDAB, and we align that to E. So that's this last alignment down here at the base of the guide tree. And putting all that together gives you the complete alignment, in principle, the best alignment, okay? But it's a greedy algorithm. So people recognize this even from the beginning, but it was the best we could do back in the 80s and 90s. Why is it greedy? Well, it's taking A and B and C and D, like the things that are most closely related to at the beginning. If that's a mistake, then that mistake, building the consensus, just percolates all the way through, and you don't end up with the optimum at the end, okay? You don't know you didn't end up with the optimum, but because it's a greedy algorithm, you should be sort of skeptical about its ability to find the optimum, okay? So that's where iterative and consistency methods came in. Muscle, Clustall Omega, MAFT, all of these use iterative and consistency methods. So what does that mean? This is the muscle example. It does all that stuff that I was just talking about, right? This step 1.123, that gets you to the previous slide, right? We've done our tree and we've done our guided progressive alignment. Then it recomputes the distance matrix based on the multiple alignment and does that thing again. So that's iterative. It's saying, I built an alignment, now rescore my weight matrix and redo the tree. Then, and you can specify how many times you want it to do that iteration. It will do it. It has a threshold for how much did the weight matrix change, and it'll stop when that weight matrix basically doesn't change anymore, or you can tell it how many times to do it. After it sort of thinks it has a good weight matrix and an alignment based on that weight matrix, now it starts chopping the tree apart and saying, okay, if I eliminate this section of the tree and only align this part, do I get consistency in the guide tree? And what that does is it allows for like sequences that really are not very similar, but they're having a big effect in the distance matrix. So if I eliminate that sequence somehow by trimming that part of the tree out and realign, in principle, I should get the same tree, or if it's affecting it a lot, I should get a better tree. If it's not affecting it at all, I get the same tree. And so I'm going to keep clipping parts out of the tree until I stop changing the result, okay? So that's the consensus part of it. So you iterate, and then you try to get to a consensus alignment, and then that ultimately is what you're going to say is your best alignment. All right, took the whole time for lecture. That's what I get for trying to put multiple and pairwise together. So we'll just kick the example to Thursday.